I would like to thank the organizers giving me the opportunity to present our recent work. This is a paper recently accepted in the PLOS Computational Biology. And it's a joint work with, mainly it's a work by Dr. Xinghua Lu, who is right now in China. And if you try to say, replace the name with a face, this is Xinghua. So we're interested in the finding the cancer pathways. The cancer, in a sense, the previous speakers already discussed, is a genomic disease because of genomic alterations, changing the function of the proteins and then causing the. And here is one picture from Augustin's paper. Is say at different rate, different type of cancer may have different rate of mutations. In addition to mutation, there is a genomic other type of genomic alterations like somatic copy number alterations and methylation. This all may affect the cancer genomic, we can say. Collectively, we call this somatic genomic alterations. So realize the importance of cancer genomics. In fact, this cancer genomics is one motivation we start sequencing human genome. And then this is in 2007, Francis Collins had a review paper on the motivated. So we need sequencing as many of the cancer genome as possible. And of course, then when the Eric Fund come over, then the TCGA and the Internet ICGC began to sequence a large number of the cancer tumors. So far available, I guess, TCGA is over 10,000 tumors. On the other side, cancer is also a disease of signaling. All these mutations reflected have to perturbing the cellular, normally well-controlled cellular signaling. When this normal controlled cellular signaling occurred, was perturbed by genomic alterations, and then that will causing the cancer. So this is just one typical, picture. People say the signal transduction pathways involved in the cell proliferation. This is epithelial. It's epidermal growth factors, and there are many proteins that involve transmitting the signals of this growth signals. And in cancers, many of these genes are mutated. Just like the previous speaker mentioned, so far, even though we know there's a long tail of that, but so far the canonical cancer pathways is incomplete. And if you look at the TCGA, on average, if you find out there are about 200 genes that is known as a driver, so-called driver. But on the average, each tumor have very few of them. But we know cancer is also a multiple pathway disease, so usually you cannot solely explain. So we are also interested in to see, can we de novo find more cancer pathways? And the approach is similar like what previous speaker said. If any of this mutation occurs, can we utilize it? Usually cells, when this signal transfer, there are some readout and some change of the cellular states. Can we combine this data to figure out what's the impact of each mutation? Can we use this one approach to reconstruct the new pathway? So it's a sort of a de novo pathway discovery. There's another characteristic of these mutation patterns. The mutations on the same pathway seldom co-occur in the individual tumor, because if you have one mutation along this pathway, usually if it is sufficient, then the rest you do not observe in the same tumor. So this is one typical uncle print draw. This is if across the row, each column is a tumor, and then if you can see this is an NFF1 gene, and in these tumors, many of they sort of have mutual exclusive. So these tumors only have one gene mutated, and this one have another one, and these are well known. They are in the same pathway. So, but you also need to know that the mutual exclusive is not absolute. This is a well-known cancer pathway. It's a PIK3CA and a P10. You begin to see in these tumors, they significantly overlaps as well. This mutual exclusivity has been used as one characteristic to try to see if we can find one set of mutations, so they tend to be mutually exclusive, there's an indication they potentially are on the same pathway. So there's a group of researchers leading, early seminal works in fact, it's the first speaker's lab from Dr. Benjamin Raphael, and also this is from the Sloan Catherine group. It's all they published this paper, so can we use the mutual exclusivity as objective function to search for sets of genes, and then to test if this set of the genes are on the same pathway. So this is this is a dendritic programs by the Raphael's group. So basically, for example, they say we try to find one set of mutation events that covers as many as tumors. If each row is a tumor and each column is one gene, and then, for example, if you find for two genes you begin to say these two genes, so maybe it's produce a better cover, and these are, if you define the k equals 3, this is their pursued objective function. You give a set of a tumor and a set of a mutation, given k, this pursue this function, trying to search for the sets. This work, they have been demonstrated this works, and they will be able to find the well-known cancer pathways. So they've also provided the novel pathways or novel candidates. But there's a limitation on this mutual exclusivity method. First, is mutual exclusivity is not a necessary, nor is a sufficient condition for the mutations to on the same pathway. I just give you the example. That is, certain pathways, there's a significant overlap, and also, as we more and more tumor get sequenced, and the previous speaker touched on this, is there's a long tail effect. So there's in the cancer genomics, so majority of the genes are mutated at very low frequency. And these low frequency ones, very easily you can find some non-overlapping or mutual exclusive sets of a relatively small k, cover a certain number of tumors. That's almost become very trivial. You can find many of those kind of sets. How to evaluate those sets is a challenging problem. And so this is so mutual exclusive metrics. Another part is a limitation of the methods I just mentioned before, is their metric does not really measure the function impact of them. It's just as how strongly these genes are mutually exclusive. As we said, this does not reflect the function impact. We're trying to have a new perspective, trying to solve this problem, is can we go to a signal-oriented pathway? So in fact, really, by definition, a signal pathway is a set of proteins or molecules that is involved transmitting a common signal. So if you can have a way to measure certain kind of signal readouts, like phenotypes, or like gene expression of things, and then you should be able to find out to see if there's the mutation pattern have anything to do with the function impact. So we formulate this as we sort of formulate this problem as this is just find a set of proteins. So when they are perturbed by mutations, they affect the common signal. And we use the TCGA data, which include the gene expression data, mutation data, and copy number alteration data. And we try to find out the function impact of mutations in terms of their impact on the gene expression changes. Then the question is, if you can find the gene expression from gene expression data, find one set of genes that are likely to be regulated by one pathway, and then you begin to find a search of the mutations that affect this gene modules. So, but it is also very challenging. You can see, if you look at these numbers, you begin to find out on each tumor, on the average, you can easily say 300 or 400 mutation events, but also thousands of differential gene expressions. So matching them is very challenging. How you found out from expression data, found out so this gene in this tumor, this particular signal is perturbed, is a challenging task. So we try to define a way to say, first, if give me a mutation data from each tumor, can I find out the genes that perform related function? And if this gene perform related function, yet if they always co-regulate in multiple tumors, then these genes potentially, you see, this whole set of genes, their change of the expression may reflect the change of the status of the particular signal in the cell. And then we try to search for this kind of thing. So we call this as a responding module. That's a module of genes that is perform related function and co-regulated in multiple tumors. So we tried to first find out the genes and that's perform related function we use in the gene ontology. I think there's no need to discuss this gene ontology for this in this audience. So we sort of have just map to the genes, differentially expressed genes to the gene ontology, and we perform a series of operations. Then we can find out trimming the graphs and merge the graphs, and eventually we can search the genes. Okay. Eventually, we try to use and summarize a set of genes so we can say, okay, this is a set of genes correlated, functionally related. We cannot collapse this anymore because then you will lose all the information. So this is and then given a set of differentially expressed genes that is perform similar function, this is a set of differentially expressed genes. And then we begin to find out in which tumor these guys are differentially expressed. And then we try to search a set of a tumor such in there's a dense connected by part of the graph, and then we can say, likely, this is one module of the genes that is densely connected to a subset of tumors. Therefore, this may reflect one signal that's perturbed in this set of a tumor. And once you have one set of you have this kind of gene expression module, then we can divide tumors according to if you have these modules that RM is differentially expressed or is not perturbed. And then we begin to find out the mutations. So here the G is become mutations instead of differentially expressed genes. We can find out the mutations that are particularly enriched in these tumors, and then yet they exhibit a mutual exclusive pattern. So this is, we believe this is a better way to find a potential pathway candidate. You may find out that there are so many of these genes, they occur the same frequency in this tumor, but they do not have as specific signal because they may co-occur in the tumors do not have this module change. So therefore their signal is not. So what we did is we represent each mutation then as a node on the graph, and we put a weight on the graph, on the node. This weight is we measure the information sort of using chi-square or the hypergeometric method to give a p-value, and then each node has the strength or the weight of the information it has with respect to a particular expression module. And then we begin to search for a set that is have a highest weight. Of course, the title is a minimal weight, right? But then you just inverse them, then you begin to search minimal weight is from an algorithm point of view. But the way we're trying to search is a set of the genes mutations that have one strongest information with respect to a specific module, and yet they show the exhibit, mutual exclusivity pattern. So if you look at this, these are the set of the genes which they provide the best coverage, and they have the strongest information, and they tend to be mutually exclusive in the tumors. I do not have the time to go through all the details, and in fact, this is a graph-based NP-hard problem, and Dr. Xinghua Lu is the expert in the algorithm design, and he designed this algorithm to be efficient. It's called a parameterization problem. These are all NP-hard problems, and it's exponential to certain kind of parameters. But if you can identify certain parameters, like a number of the genes in the module, and then he can define this as a workable, complex algorithm to search. So we can find all this, superficially, we can, anecdotally, we can find out the express modules that is involved in the common pathways. And then if we use this gene expression module to separate the tumors, we can find that the survival have a difference. So that means that these modules have biological meaning. And then we can find that there's a perturbation module, which is a candidate pathways. They have the mutual exclusive patterns, and some are on the well-known pathways. But another one, just anecdotal, you begin to see, is it systematic? And then we begin to see, does this information approach help for you to search the pathways? And then we can say, okay, we can do this experiment. Does the other well-known current existing algorithm can benefit from the signal-oriented approach? So we can find out, we try to do the experiment using the dendritic program is from Dr. Raphael's lab, and then we perform, we say if we, identify the SGA from random draw tumors, or a set of tumors have the particular RM is aberrant, or maybe we can even better is give the capability for the Dendrix to utilize the information, but we only select the top 200 genes as the input, and then we begin to see, can Dendrix identify this network that is particularly enriched in the tumors where this expression modules changed. So we can find out, yes, indeed, the Dendrix program can utilize this information to find the mutually exclusive set that's particularly enriched in the tumors which has this, modules that differentiate express. And so that is interesting. So basically, this approach can be applied to the broadly if they have other graph search algorithm. And then, of course, so what's another one contribution of our method is we have developed an efficient algorithm that is much efficient than the can find the modules it's a higher more enriched in the tumors with a particular module differentially expressed. And you need to notice this scale is in the log scale. So there's the algorithm is significant, perform much better. So this is basically a summary of our approaches. So basically, is to say we're trying to, build on existing algorithms, and we try to principles, and then we see potentially signal-oriented approach is a more biological sensible approach to search the signal pathways. And of course, our method to search the gene expression module is a start, allow you to find out the signals in the expression data. And then if you have those information, you can utilize those to search for the other candidate pathway using our algorithm or using the existing algorithm. So this is acknowledged, of course, this is a collaborative work with Dr. Xinghua Lu and Nick Namestrong. This is a funding sources. Of course, a little bit shameless. We try to recruiting people right now, and we have a Pittsburgh. We have a national NIH big data center with a causal discovery. We are one component of causal discovery is trying to find cancer pathways. So we are recruiting. And then thank you for your attention, and I guess I'll entertain your questions. Thanks.
Can take a quick question. Everyone wants to go to coffee break, huh? Well, I would just like to say real quickly that I've tried a lot of sessions and never have felt so guilty about making people have really short talks. All the talks, all 3 talks were really great and should have been like half-hour talks. So I'd like to thank the 3 speakers once again. And Xinghua will be right here, so we can go to coffee break, but you can also come up here and ask him questions.